Ale just told me that she cannot open the mic. She'll be just an spectator. So I will let Luis and Ale in. Luis is the guy who's going to join you tomorrow, who found us on Instagram. I'll turn off my camera. Ale just told me that she cannot open the mic. She'll be just an spectator.
But welcome, Ali. Welcome, Luis.
Okay, you can start.
Okay, so good evening, guys. My name is Anne, and I am a chemical engineer. I graduated at UFMA in São Luís, in the state of Maranhão. And at the beginning, I just researched about the materials, biomaterials, but I didn't give this a specific use for it. But now I am a master degree student at Unicamp, Campinas, and I am a part of the Department of Bioprocess and Materials Engineering, which is DEMBio. And now my project is related with the use of biomaterials in a biomedical field. The project is to produce a new type of wound dressing to stop bleeding using different kinds of materials. And this kind of study requires the use of some techniques like in vitro testing and animal testing. And so because of that, I am talking tonight about how can science progress without animal testing. It is possible, and how the science today use these living animals to progress and develop new kind of vaccines, drugs, and treatments in general. So during all this week, we have discussed about animals and our relationship with them, and in a different points of view. And I am a scientist, and I'm also very concerned about the use of animals. And tonight I rather to bring to you different points of view, the society point of view, and how could be harmful to the nature, to the species, use this animal, and what are the benefits of use them in this research. And at the end, I will talk about some of new alternatives that are surging now, and that could help to banish in the future the use of animals, which is a great interest for all the people in society in general, and also the scientists. And we will see, because we will see the reason behind it. Okay, so if you have any questions, you can interrupt me anytime, and I'm also will do some questions to you. Okay.
Okay.
So, animals in the history of science. Animals have been used earlier than 1957, but I choose start the history with Laika. which is a dog, Soviet dog, used in an area that is not the biomedical field. And I chose Dolly because it was very knowing the people are aware about this situation because it was revolutionary. And I will talk about the recent years and the common types of animals used in this kind of research, and why the humans use them, or the regulation involve this kind of experiments. And first of all, I would like to present you Laika. Laika was the first dog, the first living creature to be launched into Earth orbit, and in an artificial satellite created by Soviet Union during the Cold War and the space race. And she actually was sent to a suicide mission because it was always understood that she wouldn't return alive from this mission. And it happens because the pressure of the government to launch and to test these kind of experiments and how the living beings behave in this kind of situation. And the scientists didn't have the time to organize and prepare a mission for she return to Earth safely. So it was a suicide mission, and it was an example of how the use of animal exceeds the ethical boundaries, And there are two versions of the cause of head death. The first one is said by the contemporary scientists that she was kept alive for 6 or 7 days in the orbit and then euthanized with poisoned food. And then the satellite was destroyed when it was re-entering the Earth atmosphere. But another version of that said that she actually survived only about 5 or 7 hours, and after the lift of the satellite, she died of overheating and panic. And this is so cruel. And this shocked the world, and she is considered until now a sort of martyr of the Cold War. And then I will present to you Dolly, which was the first clone of an adult mammal produced by a British group of scientists. And at the time, at 1996, other animals have been cloned, but she was the first one to be cloned of an adult cell. And the purpose of the research was studying the changes that occurring on the DNA during the development of these animals when the cells became specialized in a function. It's like the truncal cells. When they are in a stage of embryonic stage, they can turn specialized in a specific function. But it was speculated the cells have not the ability to become any other different types of cells if they were taken from an adult cell. So the experiments with Dolly proved the contrary, proved that it was possible to create an animal from an adult cell. And Dolly survived during 7 years, and after her birth, he was euthanized. He was euthanized by veterinarians because she was suffering of a lung disease. So it was in 2003. And I would like to ask you if you already know about these cases and what was your point of view about this experiment.
I didn't know anything. Actually, I knew about them, but I just felt proud of science in a way.
Yes.
I didn't feel there was any kind of cruelty to them, you know.
Yeah, I think this kind of experiment that create life, because it was very serious, because humans are supposed to not create life in the laboratory, you know. And it was revolutionary, and it brings all the types of concern about this biological area of engineering and also genetics.
I see. Ale has
It's
a
funny.
comment. Go on, Luis.
Sorry. It's funny how media and television and stuff show only the bright side of these two animals. I've heard about it, Laika and Dolly, but only the success of the projects, never the pain they suffered. It's never showed on the media. I think it's very, I don't know. Science has two sides: the good sides, the improvements, but sometimes this kind of things is, I don't know. It's an ethical thing that we have to think about it. And can science move forwards without do any suffering?
Yeah, what's the price?
Yeah, what's the price, right?
Of the development and the new treatments and the new kind of drugs, and it helps us, but what the costs? What about the costs behind all of it?
Yeah. Ale has a comment here. So about Laika, every time I think of her, I feel ashamed of the humans that sent her.
Yeah.
And also she said, I think that after Laika, there was a cat that was also sent. It was super cruel. About Dolly, I'm not sure how much knowledge we had after this. It's a question she makes at the end. How much knowledge we had after this? What do you mean, Ale?
And it was interesting because the case of Dolly was just released to the public after 6 months of her birth, you know. So after that, anybody actually were aware about the situation.
Why do you think it was the case?
Sorry?
Why do you think that was the case? Why did they only release to the public after 6 months?
Because it's a very controversy experiment.
I see.
you know, fabricate life in the lab.
I see.
Can I make a point here?
Yeah, sure.
I think that the most controversial thing in Dolly's history is not creating life in laboratory, because we already did with bacteria and fungi and others kind of life. But the closer we get to the human species,
Yeah,
the ethical barrier is 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 more important. I think this is the point. If we clone a mammal, what about cloning a human being? This is the next step, the natural next step. So I think that's why Dolly was a huge deal. Still now is.
yeah, yeah. It's very controversial because of that too. Because well, when Chinese, I think about 1 year ago, said that they cloned the human in the lab. It was very released on the media, and everyone talked about it, and how could they do some kind of experiments. And the case of Dolly was to bring this kind of discussion and project the laws, and because they knew that with the release of this experiment, large amount of people would be would be not agree with that, and so they will create regulamentations that restrict this kind of experiment.
Interesting. Ali has clarified what her question was. She said, how much knowledge we had after this? And then she commented, I think that it improved our knowledge about clones. Now they're using the same knowledge to create organs for transplants, like quote unquote create. And also she said, I know what you mean, Luis. That's the point. People are already cloning their pets.
Yeah, you see, and pets is, if you think about culturally, sometimes an animal is part of our family. Some people think of your pets are their sons, as their child. So it's very interesting. And if you are about to lose your pet, then you clone them, and will keep her till you die too. It's a very dangerous thought, because what it really is important here is not the cloning stuff, but sometimes it's more cultural, it's emotional. There is an episode of Black Mirror. I don't know if you guys already seen that. When the wife clones her husband after he dies and keeps him. It's blowing my mind when I saw it for the first time. But I think it's the case here.
And the lesson of this episode is that he's not the same. Like he mimics some of the behaviors. He looks like him, but he's just not the same.
Yeah, and could never be, because they, especially when you think in levels of conscience and I don't know, personality. We can clone the biological stuff, but can we clone the personality? Can we clone all the character that makes a human being? I don't know. That's another thought that we need to develop.
Yeah, and the same goes for pets, right? When you clone your pet, it's not going to be the same one. It's going to look like it.
Of
maybe
course.
maybe it's gonna mimic better, because sometimes like animals have less ways to show their personality, I would say. So maybe you would think it's the same, but it wouldn't be. Alice said, it's 100% emotional. We have a soul, right? And Diana said, the clone will have the same DNA, but will not be the same soul, anything else.
Yeah.
Okay, I've been quite philosophical now.
Exactly.
So I think we should go on.
Okay. So, and how was the recent test with animals? Why do scientists still experiment on animals? So the first reason is that they serve as a model for human biology, because so the scientists could do this experiment to see how their bodies behave in when a drug is inserted, or a type of material, or a new kind of treatment. And they are actually a peer review of how our organism will behave in these kind of situations. And the possibility to control raising conditions, because they could make a specific diet for the animals and create them under specific conditions that will help to restrain the variables, the parameters of the process, because everything is significant for an experiment. For example, what do they eat? What do they, if they are exposed to solar light, how many times they are exposed to that, and the temperature, and all of this could be significant to the final results. So the possibility to control this rise condition, and also breed the animals, and maintain them in a specific location in the lab at the same conditions. And the third reason, there are some species like mice are genetically identical to each other. So it is important to science, because the test should be done in identical bodies, So if they are similar to each other, it's more easy to see the tendency of a treatment, or a drug, or kind of material. But because of that, more than 100 million animals were used per year today.
Only in the US, right?
Yes.
Only in the US,
Yes.
more than 100 million animals.
Only in the US, yeah. That's a lot. How many labs use this kind of animals? And so the recent tests also use specific types of species to a specific purpose. It depends on the disease that will be studied. It depends the conditions. It depends also the treatment which could be applied. And I bring this species, the zebrafish, and it is interesting because this animal can be used in different steps of life for different purpose. And this fish is used basic to the study of cancer disease, and the embryo is used to transplant cancer cells from humans to the fish. And so they have this structure that with rapid development, optical clarity, and genetically malleability, external development. And so the scientists can inject the cells into this embryo and see the first stage of the cancer cells. How do they reproduce and how do they behave in this organism? And then they can advance and accomplish with the development of the animal. The larvae possess optical clarity too, and also it's good to make image ultrasonography, in this kind of image tests that could reveal the state of the disease. And as an adult, they have the vertebral neural structure. They are low cost because it's easy and cheap to maintain them in the lab. And their genome comprises of several human genes. So they have in their genomes genes similar to ours. And so because of that, this kind of fish is used for study the development of the cancer disease. And it is also quick to breed, easy to house, and as I told you, they also care about 70% of the human genes. So they are suitable for these studies. But they suffer the disease. They suffer the cancer disease too. The other type of species which are commonly used is the rodents, which comprise rats, mice, and rabbits. They are 3/4 of all animal subjects studied in this kind of tests. They are the majority. And because they are easy to raise and breed as well, and their physiology is similar to us, and the genome is also very close to us. And because of that, they are used to modeling behaviors in toxicology tests. And for example, in my research that I am preparing a new material for wound dressing, it will be probably use a rat or a type of mice, and they will be subject to an injury, and the biologist studies how much bleed they could release and what the type of material can be effective to stop this bleeding, So in this kind of research, generally, the rats and mice and rabbits are used. And then we have the non-human primates. Like primates, sorry, like chimpanzees and monkeys, and they are very close to us. They are similar, our relative evolutionary relatives, and they are also extensively used to the scientific purpose, and they are most controversial, the use of this type of animals, because they are close to us and they look like ourselves. And then when I saw this, I thought that this is also selfish, because we don't care about the other animals because they are different from us. And how much close the animals are to us, and we have this ethical boundary and the laws to restrict the use. So the non-human primates are the less used to these tests, but it's also extensively used, and the applications are vaccines like polio, and the vaccine of polio was tested in the monkeys, and they are successful, but there is this controversial results, because the animals suffer from the disease, and we are also about to see the regulation about this kind of experiments. And I bring to you because this regulation is different from two countries, between countries. And I bring to you Brazilian laws about these tests with animals. And the first one said the animal can only be submitted to the interventions recommended before, during, and after the experiment, if it receives a special treatment established by CONSEA, which is the organ that take care of this kind of research in the use of animal for scientific purpose. And also, the animal will be euthanized on the strict obedience of this present species. So each species have a process to be euthanized, and they have to follow these steps. And it is applied, this procedure, euthanasia, is applied when the experiment is terminated, or during the phases when it is recommended, because they were under intense suffering and pain. And also, the experiments that may cause pain or distress will be developed on the adequate sedation, analgesics, and anesthesia. So here we can see that the regulation allow the suffering of these animals, and also they just regulate how they will be used, but they are not effective when it comes to apply rules about the treatment of these animals. And it could be cruel because scientists could subject the animals to a lot of pain, distress, and actually they grow under several conditions. And the regulation allows that, but this they say that it should be used analgesia and anesthesia under these conditions of pain, when the pain is not the case of study, you know. But if the case of study is the pain of the animal, it is allowed. So
Wow.
it's, yeah, it's very controversial. So what do you think about this law? I bring to you the main that I read. There are others, but these three, especially these three, came to my attention because they said that it's allowed to submit the animals to suffering and pain. Did you know about these laws?
I didn't know about these laws.
I
And
I know.
see there's a small concern about their suffering. Right? Like, oh, if they suffer too much, you are kind of required to euthanize them.
Yeah.
But as you just said, if the pain is the indicator of whether or not the experiment is working, that wouldn't apply, right?
Yeah. And this committee, which is created to regulate this, is formed by veterinarians and biomedical specialists, and also a society, a person of the society, you know.
You see.
But
Luis, you were
what
going to
I'm-
say something?
Yeah, I am thinking here. Okay, when you talk about monkeys and dogs and rabbits, but what? But there is animals that don't show signals of pain. I don't know. How do you know when a mouse is in pain? How do you know when a fish is in pain? I think that's very important because there are animals that we can see clearly when they're in pain, but and the others one.
Yes, and these laws are created based on when we feel the pain. I mean, our reflex. How do we behave when we are in pain? And most of the time, it doesn't work to this kind of animals. It could be different, right?
Yeah, I understand.
Something, right? Something you wouldn't recognize.
Yeah,
Then I have
exactly.
a comment here. It is all very disturbing. With all our knowledge, we are still making research like we did 100 years ago with animals.
Yeah. So how could experimentation become more human? These two zoologists suggest these 3R laws about the experimentation with animals. And the first one is restrict, and restrict when to use animals, you know, because every type of research that I will do in the lab, could I use animals to this purpose, or it should be more controlled and restrict the cases whether the animals would be used, you know. And the other R is refine. Is that correct, Gus? Refine.
Yes, refine.
Refine the kinds of experiment conducted them, because these kind of experiments are old. It came from the Second War, you know, and it should be more studying how to use animal, how treat them, how do the experiments, the steps of the experiment to relieve the pain that they suffer, and also the distress and the ambiental conditions to their lives. And the third one is to replace technology that becomes available. And we will see that some technologies which could be in the future replace these kind of tests with animals. So we do have other alternatives. But before I talked about the other alternatives, I would bring to you two other big issues about animal research, that it doesn't represent human patients perfectly. So we use them, but was healed in monkeys and HIV as well, but it doesn't work in the human organism, you know. So it doesn't it could not be applied in the human body. It's not effective, and it could happen. And actually, the most of the case tests in animals revealed if the treatment or the material or the drug are sorry. So this kind of tests reveal if it is safe to use this treatment, drugs or materials in the human body. So if it is safe to animals, it is safe to us. Most of the case, 19% of the case, but it doesn't show the effectiveness of the methodology or the drug or the medicine, you know. And it is also expensive. Maintain these animals in the lab is expensive, and because of that, other alternatives are created to replace these tests. The first one is computer modeling. And here we could see a vessel, a blood vessel, which was modeling through the computer modeling. And it could show how the fluids behave and what are the rate of absorption of the drug, you know. They could predict this properly. There are also in vitro tissue culture design or in vitro culture. It's based on the fact of take a culture of cells and provide specific conditions to their growth and development. And the tests could be done, for example, cytotoxical tests could be done in vitro culture precisely because we could see if the cells are dying, if the cells are growing fast, or if the cells are dying after the certain period of time, you know. And we also have the 3D tissue conglomerates. It's made by a new technique called 3D bioprinting. We usually know 3D printing, but 3D bioprinting use cells injected in a support made of a kind of biomaterial, and it allows the cell to grow and form tissues and or organs, and it could replicate the human's organs and tissues. So the medicine and the drugs could be applied on these structures, and it could be used human cells to this technique. And although all these techniques supply a new kind of treatment, a new kind of research, they also don't replicate properly the human body, but it could be an alternative. And if science persists on research this kind of alternatives, it could be done. It could be banish the tests with animals in the future, you know, by the use of one of them or combine the three of them, and it could be a new alternative to replace animals testing. So as we reduce animal testing, drug development gets cheaper, faster, and more ethical. So everyone is a winner. So guys, that was my presentation. I hope you understand my point. And if you have any questions about the last topics, you can talk to me.
Awesome. Congrats, Annie. Amazing.
Thank you.
Yeah, I've heard about bioprinting and actually how dangerous it can be as well. Like, because when technology popularizes, everyone has access to it. And nowadays, you can download the genetic code of Ebola, and you can print the virus at home if you have one of these, and that can be quite dangerous. But it's also good to know that we can use that technology to avoid suffering in other animals.
Yeah, it's also the two sides.
Yeah. Like every tool can be used for.
Yeah, it depends on the how we do use it.
And do you think there's an ethical way of using animals, or like any use of animals is should be banished?
Well, as a person, I would say that no, there is no ethical in using any kind of animal, you know, because they are living being, and we are not respect their lives, you know. We are treating them as an object to use to our purpose, because we are above them in somehow we think that we are above them. But as a scientist, I would say that science cannot be improved today without these tests.
Yeah, I was talking to Ale one of these days, and she told me that the companies that claim not to use animal testing today are basing their products on previous animal testing. So other companies or other labs or someone has tested those cosmetics, for example, on animals someday, and now that's proven, they can claim that they don't use it. But still, for the development of that cosmetic, for example, it was used at some point. And yeah, does anyone else have a question? Ale, Diana, Lucas, Luis, Vanessa.
I think I can talk right now. My nephew was here and he was screaming. I'm sorry about it. That's why I was typing. But it was amazing to see your point of view, and yeah, it was great. Congrats.
Thank you. I'm happy that you like it, and I would like to know about you. Do you think that it's ethical to use these animals for this purpose?
Well, actually, when you're gonna test like a medication for a cat or a dog, you need to use the same model. So you're gonna test in cats and dogs and cattle and whatever. So there's no how to run away from it. I really don't know, because you need to test some things, some behaviors of the drug, and some of the effectiveness. Like if it's something for flea and ticks, you need to give the tablet to the dog and then put some fleas on it and check if they're going to die, when they are going to die, after they how can I say, ingest the blood.
Yes.
Yeah. So some things you can't recreate in the lab. That's why I don't think you can avoid it.
And what about it if it's a matter of life and death? Like, I imagine, like, if one day we can stop using animals for testing, and now, for example, we are developing a COVID vaccine. It has been probably been tested in animals, or wasn't it?
Yes, I think it was, because they are still they are already testing humans, so they wouldn't testing humans without testing animals first.
I see. I've translated some papers about dentistry, and they also use rats for dentistry. Like they break their jaws on purpose and see how fast they get cured or something, and they cause some anomaly in their teeth to see how they behave, and then they euthanize it and cut their skulls in half and compare the teeth. It's quite gruesome, like to imagine having to do that. I imagine it must be a huge dilemma. Like, how do you, for example, you just showed us that you can use 3D printing, 3D bioprinting, but that's just a tissue. It's not like a set of teeth. How do you model that? Maybe in computer modeling, right?
But it could be printing also. This also structure, you know.
I see.
Yeah.
Okay, guys. So Vanessa has a comment here. I just want to thank Annie for this talk. I don't have so many things to comment, as this topic is new for me, but I like understanding the different points about animal testing. I was completely against, but I had no knowledge to found why I was against before, like to give a base to it, right? Now we know why you're against it. Louise is asking, what about the costs?
I am thinking here, how are the costs compared when you do animal testing and when you do alternatives, because we know that the industry is thinking only about money. So
Yeah.
I don't know how much more costs for you do an ethical experiment not involving animals and cruelty free, etc., etc.
Well, in case of in vitro tissue culture, is more cheaper. Is cheaper actually than
Really?
animal tests. Yeah, in vitro is very much cheaper, and the tissue, the 3D bioprinting is a new technique, so it's. I think it was expensive now. And computer modeling is also an alternative for some cases, but we could not modeling everything that happens in our body. It's cheaper, but it doesn't apply to all situations.
Okay, it's a good question, Luis, because we only the market only lines up with ethical issues and a more humane way of acting when it becomes commercially interesting. Like we only stop using fossil fuels for good when it becomes more expensive than solar power, for example. So it's always interesting to have in mind the market as well, because only thinking humanely won't do it, won't cut it. That's why Peter's article that I believe Annie read as well, that mentioned the 100 million animals in the US. Like the second section of the article shows already, like, animal testing is wasteful and unreliable. So it's proving, it's trying to prove that we cannot trust it. It's not supposed to be used by the market or by science because it's not very reliable. It's not focusing on, oh, we should stop using animals because they suffer. They show to the world, okay, this is effective at some point, but not that much. So let's try to replace for something that's more reliable. So we have to speak the language of money.
Yeah, I disagree with this statement actually, because it's not useless, but it's very limited. I would correct to this.
They didn't say it was useless, just it was wasteful.
Yeah, wasteful.
Yeah, so it causes waste, like waste of time and effort, maybe. Okay, guys, so I believe we can finish for now.
Okay.
This talk is, we are live on YouTube, so you can share the link with your friends to watch later. You can comment on YouTube, you can subscribe to our channel. I'm going to leave the link to the YouTube video right here. And thanks for coming, and thanks a lot, Annie, for your talk. It was amazing.
Thank you, guys. Thank you for coming. Annie. Have a nice weekend and see you. So, Gustavo, see you tomorrow at the Infusion meeting.
Tomorrow at Infusion.
Right?
Louise is going to meet you in person.
I don't have the address and the time for our meeting.
You do. In the form that you signed up to, there was the
oh, okay.
Name of the place and the time beside it.
Okay, I will check it out.
Okay.
Okay. Bye, guys.
Bye, guys.
Bye. Thank you.
Goodbye.
Goodbye.